The topic what we are going to discuss for today is Introduction to Paleo Botany. I'm Dr. J. Bharati, Assistant Professor, Department of Botany, Sri Sharada College for Women, Autonomous Salem. So, coming to the introduction of paleobotany, it is to be in simple terms unveiling the Earth's ancient plant life. So, paleobotany is a fascinating subject and it is a branch of botany which has been dedicated for the study of fossilized plants. It deals with the recovery, identification, and analysis of plant remains that has been preserved in the geological context that has been ranging from microscopic spores, pollen grains, to macroscopic leaves, stem, root, and the entire plant body. In essence, paleobotany acts as a historical record of plant life on Earth, allowing us to reconstruct ancient ecosystem, understanding the evolutionary history of the plant kingdom into the past climatic and environmental changes, which will help us to know how Earth has progressed and the evolution of the Earth and the evolution of plant species even today is a boon. The term paleo has originated from a Greek word palaios, which means old or ancient. As we know, botany refers to the scientific study of plants. Paleobotany literally translates the study of ancient plants. It is an inherently interdisciplinary field drawing upon knowledge on biology, especially focusing on anatomy, morphology, systematics, and ecology. And on the other side, geological studies, which include stratigraphy, sedimentology, taphonology, which is the study of fossilization process. Both of these processes stay hand in hand to learn more about what we know today. And there are reasons why we have to study ancient plants. The significance of paleobotany is very much important even today. The study of fossil plants is the wealth information that is very much critical in understanding the history of life on planet and has significant implications for various scientific disciplines. Understanding plant evolution, paleobotany provides a direct evidence of how plants have evolved over millions of years. We also examine the morphological characters, anatomical characters, reproductive structures of fossilized plants from different geological periods where we trace the origin of major plant groups which has inhabited the whole earth for years together. For example, pteridophytes, which include ferns, gymnosperms, angiosperms, these have been fossilized specimens for years together and it is an hidden treasure even today. Identifying transitional forms and understand the development of key innovations such as vascular tissues, seeds and flowers gives us an idea how nature has evolved today. Reconstruction of ancient ecosystem, paleoecology, fossil plants assemblages found in specific rock layers offer a snapshot for vegetation that exists in the particular place and time. By analyzing the type and diversity of fossil plants, paleobotanists can reconstruct ancient habitats, understanding plant-animal interaction and interfere the ecological conditions that have prevailed. Interfering past climates, that is paleoclimatology. Plants are very sensitive to environmental factors. Even today, we find plants as ecological indicators, such as these environmental conditions such as temperature, precipitation, and atmospheric composition are very much important to know about the plants and the sensitivity. Certain morphological features of fossilized leaves, example their size, shape, presence of teeth, and the distribution of specific plant group can provide us valuable clues about the past climates. This information is crucial for understanding long-term climatic change patterns and their impact on life. So, what is biostratification and geological dating? Plant fossils, particularly spores and pollens, can be abundant and geographically widespread in sedimented rocks. These evolutionary changes over time makes them an excellent index for fossil for correlating rock strata from different locations and determining their relative ages. Understanding the history of life, plants are the primary producers, forming the base of most terrestrial and many aquatic food webs. Their evolution and diversification has profoundly influenced the evolution of other life forms that includes animals and fungi. Paleo Paleontologist data or paleobotanical data is therefore essential to understand the broader history of life on Earth. So, economic applications, paleobotany has practical applications, particularly in the exploration for fossil fuels such as coal and petroleum that we even use today. For example, the coal formations from the compressed and altered remains of ancient swamp vegetation and the presence of certain plant fossils can indicate environmental favorable for formation of oil and gas deposits under the earth. Paleobotanical data can also be used in the study of understanding how human and ancient population utilized plants for their daily living. What do paleobotanists study? The types of plant fossils, how they are preserved, how they can be, how they are utilized even today is an interesting part of this study where you can identify different varieties of fossils being preserved in from different ages on different stratas. To look forward, impression and compression. These are typically two-dimensional fossils formed when plant parts are flattened between layers of sediments. So it is very clear that you can find the complete structure of the plant or the complete impression of the plant on the sedimented layers. Coming to impression, impression are the outline of surface features that are left behind, while compression retains organic materials often as carboniferous films which can sometimes be chemically analyzed. So the difference between impression is the outline, what we find will be the impression, and compression is that due to high pressure there are some organic materials that have been left over on the strata which have been in the form of carboniferous film, and sometimes these can be chemically analyzed to know about further data and information that is to be collected. Coming to petrifaction or pre-mineralization. These are three-dimensional fossils where plant tissue are infiltrated and replaced by minerals. This is one of the interesting study where when we go to the past, what happens is that there may be a loss of very big diversity where most of the plant tissues have been damaged. When it comes to years together, what happens is that there is a transitional change where the, the mineralization process gets converted. Here, all the plant tissues has been replaced by minerals such as silica, calcium carbonate, iron pyrite from the surrounding groundwater system. So there is a replacement of minerals. Petrifaction often preserves intricate cellular details, allowing details of anatomical studies. Petrified woods are the common examples which we can find in most of the preserved places or the fossilized museums. Mold, next coming to the molds and cast. Mold is a negative imprint of the plant part left in the sediment after the organic materials has decayed. So you can, for example, not just the plant parts, there are also animal molds that you can find where the organic material is decayed, but it has the outer structure. And the next is the cast is formed when the mold is subsequently filled with sediments, creating a positive replica of the original plant structure. So some are called as unaltered remains. In rare cases, plant material can be preserved in its original or near original state, such as leaves frozen in ice, plant fragments preserved in amber, or peat deposits. These are called as unaltered remains. You can find the whole structure of the plant in amber. For example, if a pollen or a small insect or the smallest flower can be preserved in amber, and the full unaltered remains have been found for further analytical studies. Spores and pollens. These microscopic reproductive structures are highly resistant to decay and are often found in abundance in sediments. Palynology, the study of spores and pollens, provide crucial information about the vegetation of the past and widely used in biostratigraphy and paleo-environmental reconstruction. Coming to phytoliths, these are microscopic silica bodies produced by some plants within their cells. Phytoliths are resistant to decomposition and can persist in soil and sediments long after the rest of plant has decayed. Their morphology can often be used to identify the plant groups and even species, making them valuable in paleoecology and archaeology. And these microscopic silica bodies produced are within their cells. Coming to chemical fossils, these are otherwise called as biomarkers. These are preserved organic molecules that were produced by ancient plants. Analyzing these chemical structures can provide insights into the type of plant that were present and their metabolic processes even when macroscopic processes are absent. Also coming to paleobotanist study fossils and their methods and techniques. These are completely related to field works and then to the dry labs. So coming to the field work, location and excavating fossil bearing rock layers is a crucial first step. First, identifying the place, identifying the region and identifying any fossil from the depth of the earth is a tough task, and in the excavating process identifying rock layers is the crucial and the first step. These involve geological mapping, careful removal of rocks, and documentation of context in which fossils are found. Macroscopic examination. Larger fossils are cleaned, prepared, and examined with naked eyes or using hand lenses and light microscope to study the overall morphology. So macroscopic examination is what we see naked. with naked eyes, when you identify there's some impression, compression, mold, some formations, and very elegantly taking up these fossils, identifying it and giving it a raw data for the first time how this has been preserved. Next is the microscopy. Thin section of the petrified material or macerated compression are examined under light microscope to study the cellular anatomy. So knowing the anatomy is most important to know the evolutionary process. For the next step, SEM analysis or scanning electron microscopy and TEM analysis, transition electron microscopy, provides even higher resolution images of fossilized structures. Paleopalynology. The sediments are proceeded to extract spores and pollens, which are then identified and counted under a microscope to determine the composition of the past vegetation. So if any spores or pollens has been identified, the count of the pollen or the count of the spores is being done just for and raw data to know about the plant structure. Next is the chemical analysis. Technique like GCMS, gas chromatography and mass spectroscopy, and pyrolysis, gas chromatography and mass spectrometry are used to analyze the chemical composition of the fossil plants as it has been identified as biomarkers. Next is the radiometric dating. The methods like carbon 14 for younger fossils, potassium, argon or uranium lead dating for older rocks containing fossils are used to determine the absolute age of fossil remains. Even today, one of the advanced techniques what we see is that the development of chemical analysis, radiometry, and the high technology what we see today is identification has become a bit simpler when compared to the past. Here, analyzing using carbon 14 is one of the best way to identify the age of the fossil. As we know, the geological calendar or geological time scale is an everyday Earth's calendar which adds up data where radiometric techniques adds up more data to in addition of new species, new identification and new fossils. Comparative morphology and anatomy. Fossil plants are compared with living plants to identify similarities and differences, aiding in their classification and understanding their evolutionary relationship. Phylogenetic analysis. Modern computational methods are used to analyze morphological, anatomical, and molecular data from fossils and living plants to reconstruct evolutionary trees. So, constructing an evolutionary tree is one of the base structure where we can identify every changes and every evolutionary developments that has been happened and happening in the earth even today. Paleoecological reconstruction. By analyzing the type of fossil plants found together, their abundance and associated geological evidence, paleobotanists reconstructed ancient environments that includes vegetative types, climate, and potential interactions that happens between organisms. So coming to the conclusion, a window to green history of Earth. Paleobotany offers a unique and indispensable window into the history of plant life on Earth. By studying the preserved remains of ancient flora, we gain a deeper understanding of the process that has shaped the plant kingdom, the evolution of terrestrial ecosystem, and dynamic interplay that happens between life and environment over geological timescales. As we face current environmental challenges, the insight of paleobotanical research can also provide valuable context for understanding long-term environmental changes and resilience the ability of plant life.